systems, while emulsions are a milk system, are a biphasic system with a very big droplet of the inside phase. We developed different macroemulsions to improve the solubility both of elanoic acid and an extract, enriched extract of pentacyclic triterpenes that contain a very high amount of elanoic acid. Both elanoic acid and extract are very soluble matrix because the solubility of elanoic acid in water is less than 1 microgram per milliliter, and the extract has a solubility about 7 microgram per milliliter. We used different macroemulsions and improved the solubility of elanoic acid or extract because they increase the solubility in the oil due to the presence of the oil at the tenside. We developed 6 macroemulsions containing elanoic acid between 1 and 3 milligram. It also the solubility of extract has been increased until 3 milligram per milliliter. Then very good results for the solubility. In this slide, I show you the composition of different macroemulsions. To select the component of the macroemulsions, in particular lipophilic phase and tensite, we used compounds applied on the market for food application, for oral application, oral administration, and substances considered by FDA as grace, then this means generally recognized as safe. We performed 4 types of macroemulsions containing the first macroemulsions in blue. Campomol PG8 is the oil phase. Transcutol twin are tensites. Another macroemulsions able to solubilize until 3 milligram of olenoic acid or extract containing Nigella oils, Nigella oil and isopropyl myristate as oil phases, a transcutol cremophar tenside. The last one, macroemulsions, contain transcutol HUP as lipophilic phase, TPGS and capriol as tenside. TPGS is a derivative of vitamin E, and similar to transcutol, campomol, or cremophor, these compounds are also penetration enhancer, then are used in the formulations to improve the permeability across intestinal membrane. Macroemulsions can be marketed as solutions, or it's possible produce the systems without water, then the SMETs, to insert these SMETs inside a soft gelatin capsules. We characterize by dynamic light scattering technique and also the whole microscopic technique and chromatographic technique, the macroemulsions, to evaluate the size of the droplets of the inner phase, the homogeneity of the system, and also the load capacity of the formulation. And then we performed also stability study. Stability of the formulations during the time is very important for market applications. We studied the stability of the macroemulsions at 25 degrees centigrade and 4 degrees centigrade, and we evaluated the physical and the chemical stability. And all formulation resulted stable for 8 weeks. The formulation also are considered regarding the stability in gastrointestinal fluids, and then we produce a simulated gastrointestinal fluid. We tested the stability of the formulations, and the formulations are stable in this environment. Also, we considered the release of elanoic acid extract and also of the extract from the macroemulsions. I show you here 2 results about campomol and Nigella oil. In this case, we obtain a prolonged release of elanoic acid. The formulations improve the amount of elanoic acid released from the formulation respect to the solution. This is a saturated solution of elanoic acid. Also, here the results about the extract. In this case, the macroemulsion realizes a prolonged release of the extract with respect to the solutions. Pampa is a parallel artificial membrane permeability assay, is an in vitro test to evaluate the increase of permeability of the compounds due to the formulations. We have a donor compartment, an acceptor compartment, and here a membrane that contains lecithin and cholesterol to mimic an intestinal barrier. Oleanoic acid that also extract are impermeable, not permeable compounds and matrices, but in the presence of the macroemulsions, then the formulations and the constituents of the formulations, in all cases improve the permeability across Pampa membrane of the oleanoic acid or pentachylate terpene. This is a good result, not only for the solubility, but also for the permeability across the membrane. Another nanocarrier nanoformulations are developed for OLEAF4VALUE. OLEAF4VALUE is an extract enriched in aleuropein, then enriched extract containing polyphenols, and the polymeric micelles are colloidal system aggregate constituted by alphaphilic polymers. In this case, we tested different amphiphilic polymers to improve the solubility, but particularly also stability of OLEAF4VALUE. We select in particular poloxamel or pluronix that are amphiphilic polymers constituted by ethylene oxide as hydrophilic portion and propylene oxide as lipophilic portions, and TPGS is a derivative of vitamin E used also in the macroemulsions. The best system resulted ternary polymeric micelle constituted by pluronix L121, pluronic P188, TPGS in the weight ratio 2:1. In this micelle, in this system, we obtain a solubilization of 50 mg per milliliter of extract. The micelle have a good physical and chemical characteristic by the size, the homogeneity, and also the efficient encapsulation efficacy. We study the system also after freeze drying process, because to obtain a solid product useful for market application, also for formulation, for example, in capsules or in tablets. Also in this case, after freeze drying product, the micelles maintains the same good characteristic such as dimensions and also encapsulation efficacy of the extract. We performed the in vitro release study in PBS. We obtain a prolonged release of the extract from the micelles, and in the case of a Pampa experiment, we obtain a very good increase during the time of the permeability of OLEAF4VALUE of the extract. To improve the stability of the OLEAF4VALUE, we performed the stability study at 4 degrees centigrade on a colloidal dispersion, and then in a polymeric micelle, in this case as in liquid form, or as a freeze-dried product, a solid form. As colloidal dispersion, the micelles remain stable until 7 days, only for 1 week, and after we have an increase of size, a decrease of polydispersed index, and a decrease of encapsulation efficacy. Encapsulation efficacy is related to the stability of the extract. While in the case of a freeze-dried product, and then considering the solid product, we obtain a good result in size until 14 days, then 2 weeks, and the system, the OLEAF4VALUE remains stable for 2 weeks. Then the encapsulation efficacy remains stable during this period of the test as a solid, if we consider the formulations as solid formulations. About macro formulations, we developed 2 types of formulations, solid dispersion and recently the co-ground products. Solid dispersion is the dispersion on a hydrophobic drug or active ingredients in hydrophilic carrier to improve the solubility due to the amorphization of the drug inside the hydrophilic polymers. We can use it different preparation method. In this work, in this project, we developed 6 formulations to improve solubility both of lanoic acid or extract, and we used two hydrophilic excipient or poloxamer, P4 poloxamer 407 or 188 or gamma cyclodestrins. This is a figure that represents the method for preparation of solid dispersion. We mix in a mortar polymer and olenoic acid. We obtain a physical mixture. This is our reference formulations. Then we put some drops of ethanol inside these physical mixtures and we blend in mortar. We obtain a kneading product. Or we use a solvent evaporation method. We mix polymer with olenoic acid and we add ethanol. We put under magnetic stirring for 2-3 hours and then we evaporated the solvent under vacuum. We tested the different polymers and different molar weight ratio between solid and olenoic acid and hydrophilic polymer, and we performed the solubility study to evidence the influence of hydrophilic polymers on the solubility of lanoic acid. Then the best solid dispersion was characterized by solid techniques such as X-ray, SIM, infrared, differential scanning, calorimetry. About the solubility, we have in the case of solid dispersion regarding the olenoic acid, a very big increase of solubility with both with the kneading method as solvent evaporation method, in particular for 2-poloxamer and gamma cyclodestrin. I remember that olenoic acid is a very insoluble molecules. The X-ray and the water technique demonstrate that we have in green there is the X-ray of solid dispersion that improve of the solubility is due to the amorphization of lanoic acid inside the polymer. Taking into account, considering a possible market application, we insert the solid dispersion in a hard gelatin capsule and we perform the solubility test. We show you the results of the 3 polymers selected to improve the biopharmaceutical characteristic of lanoic acid, and in all cases we have an improvement of the dissolution profiles of lanoic acid. In black, it is in black here this line also this represent the solubility of the dissolution profile of lanoic acid alone. The other lines are the dissolution profiles of lanoic acid from solid dispersion obtained with a different method. The same test was performed considering the extract, not only lanoic acid, and also in this case this is the dissolution profile of extract. With solid dispersion we obtain a very good increase of the solution profile of the extract. About the permeability, also in this case we have a very good improvement of the permeability across pampa, because after 1 hour, in particular after 2 hours of the test, all permeability, the permeability of the extract or lanoic acid from the solid dispersion resulted improved. The last one, macro formulations, is a formulation obtained by mechanochemical activation. We use a mill ball and we put inside this mill olenoic acid and a stabilizer. The energy applied by the mill produces a morphization of the lanoic acid, a reduction of the size, and also an interaction between lanoic acid and stabilizer. We tested a different stabilizer, evaluated the influence of the milling process and the stabilizer on the solubility of the lanoic acid. We tested also different weight ratio lanoic acid stabilizer and different milling time. We have the best result with sodium cholate. We tested 1-to-1 weight ratio and 1-to-2 weight ratio between the lanoic acid and stabilizer, and 2 time milling time 30 or 60 minutes. We obtain a good results because we have a very high improvement of solubility of lanoic acid. Lanoic acid has a solubility less than 1 microgram per milliliter. We have about 700 microgram per milliliter. Also with the results, not too high results, high values with hydroxypropyl methyl cellulose, polyvinyl pyrrolidone. Also we considered the physical mixture of lanoic acid sodium cholate to evaluate if the to prove if the improvement of the solubility is due to the process, the milling process. In fact, the physical mixture with a physical mixture with sodium cholate, we have an improvement, only a small improvement of solubility of lanoic acid until 6 milligram per milliliter. Then high improvement is due to the energy applied by the mill process. In this case, we produce the capsules, our gelatin capsules, and I show you the very good improvement in the solution profiles of olanoic acid. This is the olanoic acid alone in the capsule. This is the dissolution profile of co-ground mixture put inside the capsule. We have a very good improvement of the dissolution profiles. And this dissolution profile is higher than also the dissolution profiles of solid dispersion. Then this is the best formulation obtained until now to improve the biopharmaceutical characteristic of the olanoic acid. This is all for my part, and thank you for your attention.
Thank you very much, Camille, for this complete presentation and the influence of the optimized formulation on permeability and solubility. Now for our next workshop, Dr. Christian Bucher, will you be so kind to share your screen in presentation mode? I would like to remind you there is a follow-up workshop starting at 10 as well, and also that this meeting is being recorded for all of you that have joined in the last minute. Yeah, so please, Christian.
So can you see it?
Yeah, we are seeing your screen now. Please, Christian, the floor is yours. Thank you.
Yeah, thank you for having me here. At first, I'm a postdoctoral researcher in the group of Professor Bethjen here in Halle at the University of Halle. And I want to present you some general information about the research we are doing here. And for this, we are using the tiny nematode Caenorhabditis elegans. And we're interested in the study of natural compounds. And as a little information about C. elegans. If you didn't already hear about it, it's a tiny nematode, round about 1 millimeter in size. It has some advantages as a model organism. For example, it has a very short life cycle of just about 3 days. And what's also very interesting is, especially for aging research, that C. elegans has a very short lifespan of around 3 weeks. And that's closer to some unicellular organisms, model organisms like yeast, than other more complex organisms like Drosophila or mice. C. elegans shares some features with more complex organisms. So C. elegans has a nervous system. It has muscles, an intestine, and so forth. But also very important organs and tissues are missing. For example, a distinct brain is missing, or cardiovascular system, a liver, just to say a few. So why is it still a very suitable model organism to or for the research on the effects of natural compounds? It is because C. elegans shares a lot of evolutionary conserved signaling pathways, like for example here the insulin IGF-1-like signaling pathway. And so our main goal is to investigate the molecular mechanisms of natural compounds if they are, or if they induce some positive or protective effects. And for example, here in this signaling pathway, there's in the end the transcription factor DAF16. And here in our lab, for example, we can use the transgenic strain of C. elegans that expresses the DAF16 transcription factor that is tagged with the green fluorescent protein. And so it's quite easily visible via fluorescence microscopy if we modulate this signaling pathway with some phytochemicals. Further, if we find some compounds that have protective effects in the wild type, we use, for example, loss of function mutant strains to investigate the involvement of these signaling molecules on the protective effects we have found. So we use some different experiments to, for example, investigate the influence on intracellular accumulation of reactive oxygen species. We look for protective effects against several kinds of stress, or as mentioned before, what's quite fast possible is the influence on the lifespan of the animals. But also we are interested in possible protective effects in very simple models of human neurodegenerative diseases. For example, like Alzheimer's disease or Parkinson's disease. And I want to show you some results we have generated with an extract that contains a certain amount of xylo-oligosaccharides or hemicellulose. And here on the left side, you can see the resistance against a thermal stress. And here we found that the highest concentration of this extract, that's called XOS, induced a protective effect in this assay. And on the right side, it's called the DCF assay, where we measure the intracellular generation of reactive oxygen species. And here we also found that this extract has antioxidant activity in our in vivo model. Further, in the so-called paraquat experiment, paraquat is a herbicide that generates reactive oxygen species in the cells of the animals, and this leads to the death of the animals over time. And here we also found that this extract in certain concentrations protected the animals for a certain amount of time. And lastly, here on the right side, you can see lifespan experiments in the wild type, where one concentration of this extract induced a prolongation of the lifespan of the nematodes. In the second part, I wanted to show you two models for neurodegenerative diseases that we can use in our lab. And at first, it's a model, a very simple model of Alzheimer's disease. In this model, the nematodes express the human amyloid beta peptide in the muscle cells, in the body wall muscle. And this leads to paralysis over time. As you can see here, within 8 hours after we start counting this, the animals are nearly complete paralyzed, or the amount of animals that is on the plates. And unfortunately, when we tested 2 extracts enriched in oleuropein, or what we have previously heard, the pentacyclic triterpenes, we only found a minor delay in the onset of the paralysis. However, in another model, that is a model for the hereditary disease Chorea-Huntington, we also used a transgenic strain, and this strain expresses a mutated version of the human huntingtin protein that tends to aggregate, and over the time of around 10 days, the neurons, these are touch-sensitive neurons, are degenerating. And these neurons also express the green fluorescent protein that we are able to visualize it with fluorescence microscopy. And then we can count the amount of animals showing defects or intact neurons. And here we found that the pentacyclic triterpene enriched extract rescued these neurons from degeneration, compared to another extract containing oleuropein, which had no such effect. And in this model, what also very interesting is that, as I mentioned, these neurons are touch-sensitive neurons, so we also can see the physiologic outcome of the rescued neurons. Normally, when you touch the head of C. elegans, it wants to move backwards, and this backward movement is countable. And here we also found that the extract containing pentacyclic triterpenes rescued this the touch sensitivity of these animals. And with this, I want to conclude very short that we are using a well-established model organism to investigate the effects of phytochemicals. We use a variety of experiments to investigate the modulation of oxidative or thermal stress. We investigate the influence on lifespan, on aging, and especially we're interested in the molecular mechanisms that lie behind these protective effects of phytochemicals. But we also have to remind us always that these results we generate are just preclinical results, and that it's of course not directly translatable to humans. With this, I want to thank you for your attention, and please feel free to ask questions.
Thank you very much, Christian. It's really impressive, these results of the phytochemicals into the different molecular mechanisms in these model species. Now, Dr. Thorsen, would you be so kind to share your presentation in presentation mode? He is a researcher at NITO Food Lab facilities in the Netherlands. They have been hosting us recently in a fabulous General Assembly. So yeah, that's perfect. Thorsen, please, the floor is yours.
Thank you very much. Yeah, what you said already, at NISO, we are focusing on food, or how its nutrients can basically affect the human organism. And in the last couple of decades, we are focusing really on the intestinal tract. So we look at the gut microbiota, so the intestinal organism, microorganism, how they can metabolize nutrients and how it can be absorbed. So we also look at the intestinal epithelial cells and also the immune cells. And in this project, we look specifically at the extracts from the OLEAF leaves, how they can affect the gut microbiota, but also the epithelial cells and the immune cells. So the first couple of slides, I'm going to focus on our microcolon model, which is an in vitro system where we use human microbiota, so microorganisms from fecal samples from human individuals. We culture it anaerobically, which means we culture it without any oxygen. We have specific anaerobic chambers for that, and we do it in a high throughput fashion. So we usually use 96 well plates, or if we do other analysis, then we use smaller formats, but usually it's like a high throughput assay. So you can use a lot of different extracts, for example, from the OLEAF leaves, and can screen it and see how the microbiota composition is developing over this incubation period, but also which kind of metabolites the microbiota is producing. And then afterwards, after this assay, we separate the bacteria from the supernatant, and then in the bacteria pellet, we can look at the gut microbiota composition if it changes after the 20 hours of fermentation period. And in the supernatant, we can look at the metabolites. Usually we do short-chain fatty acids, but here we also look at different phenolic components and the metabolites, which I'm going to show one slide. But we can also use this supernatant to incubate our epithelial cells with these fermented products, but that for later. So first slide, just about the gut microbiota composition. Here in this slide, we did first the qPCR, but we also sequenced already the gut microbiota, but I don't have the results here yet. And the advantage of this qPCR is you can basically quantify different groups of bacteria, different strains. For example, here we had primers for Clostridium difficile and for Bifidobacterium, and we looked how this OPA20 or OPA70, which are these OLEAF extracts, how they affect the gut microbiota, these specific strains. And for the Bifidobacterium, which is known to be a beneficial bacteria, it can produce short-chain fatty acids, which we can use the host as nutrient or like energy sources, but it's also important for the epithelial cells for different pathways. And for the Bifidobacterium, we didn't see any effect. Neither the OPA20 or the OPA70 really showed like a strong effect on the abundance of this Bifidobacterium. But if we look at the Clostridium, which is a known pathogen, which can induce diarrhea in humans, we saw that especially the OPA20 was a nice almost dose response in reduction. So the more OPA there is, also the more Clostridium difficile was reducing in abundance. But this qPCR approach is a very targeted approach. You can do quite a lot of primers, but you never really grasp the whole microbiota composition. And this is why we have data from the 16S, which is also a specific gene of bacteria, where you can basically get the whole microbiota composition. And there we didn't really see also much changes. But now we also have metagenomic sequencing, which uses shotgun sequencing approach. And there we can also look at the function and the composition. So our bioinformaticians are working hard on it to get this data. But so far, it looks like that only specific bacteria seems to be affected by this OLEAF extract treatment. But these bacteria that can metabolize these phenols. So one of the biggest polyphenols in this OPA extract is oleuropein. And from the literature, we know already that there are some metabolites. For example, oleuropane can be metabolized in hydroxytyrosol via the gut microbiota. So we set up an MS method where we can basically at the same time measure different polyphenols and also different metabolites from these polyphenols. And here I'm focusing on the OPA20. So we took samples at 0 hours, so at baseline, at 6 hours of fermentation in our microcolon model, and also at 20 hours of fermentation. And you see the oleuropane is in dark green. At 0 hours and 6 hours, there's not much change. So it's like around 300,000 microgram per kilogram. But then at 20 hours, if you look at the axis, it's only like around 6,000 microgram per kilogram. So it reduces a lot. That means that the gut microbiota can metabolize this polyphenol quite efficiently. At 20 hours already, it's like almost gone. And if you look at different metabolites, for example, tyrosol, you can see nicely coming up already at 6 hours, it increases, and at 20 hours, even a little bit more. So we found in this MS approach a couple of metabolites which appear to be specific for this oleuropane or for these polyphenols, which are metabolized by the gut microbiota. And then what I said already, we use these microcolon supernatants, and we incubate with epithelial cells. For that, we use CACO2 cell line, which is a cancer cell line of the intestinal epithelium. And you can grow these intestinal cells on a filter, which is connected to another cell line, which is a THB1 cell line. These are monocytes, and you can differentiate them into macrophages. So basically, we can mimic the intestine. We can put in the apical site, which is the top layer, we can put either the unfermented olive extracts or the fermented olive extracts on top. They are in direct contact with the epithelial cell, so the CACO2 cell line, and they are covering a filter. So usually this epithelial layer, also in human conditions, in vivo conditions, it's very tight. Most components, they cannot pass it. But if you disrupt it, for example, in this system, we use interferon gamma and lipopolysaccharide, which induces inflammation and basically makes this epithelial layer more porous. So also these metabolites, they can go into the basolateral sites, they can pass this filter, and they can interact with the immune cells. So with this project, we wanted to see if this OPA, like this olive extract, can basically recover this inflammation, this disruption of the system. So this slide shows you the results for the OPA20, where I showed you already some metabolites. So we used unfermented one, which is the pink line, which is continuous, and we use 5 milligrams per milliliter. And if you compare it to the black line, which is the lowest one, which is the interferon gamma LPS incubated, and if you look at the value, the trans epithelial electrical resistance, which is a value showing how permeable the epithelial layer is. So the lower the value, if it goes towards minus 100, also the more permeable the epithelial layer is. So you can nicely see when we induce inflammation, this permeability increases. There's more metabolites basically passing through the epithelial layer. But if you use already the OPA unfermented, you can see the values are much higher. So the unfermented polyphenols, they can already improve the epithelial barrier function. And if you look at the fermented one, we have at 2 different concentrations. So it looks like there's some kind of dose response. You can also nicely see that these tier values, they go up. So it's also recovering the intestinal barrier function. And then we also included a water control, because we know already that from the media itself, these microorganisms, they can convert nutrients from the media also into, for example, short-chain fatty acids. So that has already an effect. But still, all the OPA components, they're still higher, at least at 48 hours of fermentation, higher than the water control. And then we also used another OLEAF extract, which has 70% of oleuropane, so a higher concentration of oleuropane. And there it was interesting when we look at the unfermented one, which is the continuous red line. It's very similar to the inflammatory condition, which suggests that at 70% oleuropane, this concentration is too high for the system. So it also basically this can disrupt some kind of the epithelial barrier function. But if you look at the fermented components, which are the dashed lines, you can also see again, similar to the OPA20, that it's higher than the water control. And there's also some kind of dose response. If you look at 0.5 milligrams compared to 5 milligrams, which is the highest graph, you can see that it nicely recovers the intestinal barrier function. So these, especially these metabolites produced from these polyphenols, they improve intestinal barrier function. They can recover from this inflammatory induced, which is very interesting later if we have an OLEAF extract and we can use it as some kind of, not necessarily treatment, but at least we can improve in human individuals the intestinal barrier function. So that was my presentation. So I'm happy to answer any questions.
Very much, Thorsten, for the results on the microcolumn and the microbial composition. It's really amazing how you can replicate and interesting. So for the next part will be the questions and answer part, which Andrea Leon, my co-worker at Innovarum, will host. So Andrea, would you be so kind to share the screen with the questions?
Yes, so we will be having an interactive question and answer section. And after we finish with the questions and the answers, if anybody has doubts or has any other open questions that you would want to ask, feel free to ask our
Andrea,
speakers.
I think the audio is super bad. I don't know what's happening. It's kind of
you're
echo.
right. You're right, Andrea. Maybe you should get closer to your microphone, or try to move it. Maybe you make a test, please.
Yes, sure. How about now? Do you hear me?
Yeah, I
better?
think maybe you should try to choose another setting for your micro because the connection is quite bad.
Does it work better? Or... Okay. Tienes.
Ciérralo, páralo. We're trying to solve this problem with my computer.
Can you hear me better right now?
Yes.
Yeah, perfect.
Yes.
Okay. Okay, so I'm gonna share my screen through here. Okay, are you able to see my screen?
Yeah.
Yes.
Okay, perfect. Okay, so as I was saying, and this will be an interactive question and answer session. And if you have specific questions to the speakers, after we collect your answers, then you will be free to ask any open questions that you might have. Okay, so now you can scan the QR code to access the live questions. Perfect. Are you ready? Okay. Perfect. So this is the first question that we have. What is the purpose of using micro and nano drug delivery systems? And this is related to Professor Camila Bergonzi's presentation. So
Yes,
you can type your short answer. And yeah, Camila, you could explain probably more details.
Yes. Can you hear me? It's okay?
Yes.
Okay. Then the application of drug delivery systems has the aim to first improve the solubility of substances or of the extract of different complex matrices. Another aspect is improve the stability of the drug loaded into the formulations, and for pharmaceutical application it also important improve the solubility also to reduce the dose of a drug, for example, and reduce the side effect due to the related to the drug. It's possible also derivatize the surface of the micro and nano carrier, in particular nano drug delivery systems such as liposome nanoparticles, also to obtain a targeting of the action, and then action related to a particular organs, cell, tissue, and reduce the side effect due to the interaction with all cells. Other aspect, important aspect is the development nano drug delivery systems also to prolong the release or obtain a modified release with respect to conventional formulations. It's important to select the component of the formulations to improve the safety, also the carrier, the nano drug delivery system. And I'll show in my presentation, we selected all excipient used in the food or drug applications on the market, because sometimes on polymers or some tenside could have a toxicity for the application in the human. The toxicity also related to route of administration, for example, the topical application, parenteral application, the toxicity is also different between one component and another component selected for the formulations.
Okay, perfect. Thank you very much,
You're
Camila.
welcome. You're welcome.
Now we can move to the next question related to Dr. Christian Butcher's presentation. So the question says, why is the nematode C. elegans a suitable model organism for the initial study of natural compounds? And you have 3 options of answers, and you can select your preferred choice, and then we will see the correct answer. Are you all able to vote on and participate? Okay. Okay, Christian, I don't know if you would like to explain a little bit the answer for this question.
Yeah. So the 2 people that have voted are correct. Yeah, the main reason C. elegans is a quite good first model organism to investigate the effects of natural compounds, because important signaling pathways that are related to, for example, aging or diseases are evolutionarily conserved. And for this reason, we can investigate modulatory effects of these compounds on these signaling pathways. But the outcome is, of course, not always the same. So, for example, the insulin IGF-1 signaling like signaling pathway in C. elegans is related to aging, for example. And when you inhibit this signaling completely, the worms live twice as long. But you can't really translate it directly to humans, because if you inhibit the insulin signaling or the receptor, the insulin receptor in humans, you don't live twice as long, but you get diabetes. But we focus then more on the transcription factors that are involved. If they are, how we can modulate them and bring them or bring the cells to increase their self-protection. So I hope this was a little bit informative. Okay.
Perfect. Thank you very much for your answer and your explanations. Okay, so that is it. We only had 2 interactive questions. So in case anybody else has any other open questions that you would like to ask the speakers, the floor is yours. Anybody who would like to ask questions? If not, then we will close the session for today. Okay, so no questions as I can see. So yeah, thank you very much to our speakers and for everybody else who joined. And we hope to see you very soon in another session of the OLEAF4VALUE workshops. We have another workshop going on in literally 2 minutes. So if you would like to join, we are posting in social media the link to the event. So yeah, you can just hop on the other call and we would love to have you there. So thank you very much for joining us. If you have other questions, you can always email us and our speakers will be happy to assist you if you have any questions. So thank you very much and we hope to see you soon in another workshop for OLEAF4VALUE.
Thank you.
Thank you. Bye-bye.
Bye.
Thank you.
Ciao.
Bye-bye.